Alright guys, time to start on chapter 11. This is one that a lot of folks have trouble with, understood, but we've got to go through it. I'll try to go kind of slow, and you guys make sure you write down the questions that you don't understand. Remember, you're gonna have an exam on this on the 25th, so it's not very far away. Okay, going into it. Genetic material. Well, we've got our DNA and RNA. We move the genetic material or we use it by transcription. So we've got our DNA in the nucleus, or if it's prokaryote in the nucleoid region. We know we've got different nucleotides on there. So we make an RNA copy of that. So if this is A, and this is T, and this is C, and this one is G, then I can make an RNA copy by using U, A, G, C. So all we do is we use the complementary nucleotide. We know that A always goes with T in DNA, and A always goes with U in RNA. Make sure I mark that so there's no question about what we're talking. And the same thing with C and G, they always go together. So this is how we do a transcription. We copy the information from the DNA and we use it to make our proteins. This RNA is then going to be translated. Polypeptides or proteins are synthesized from the RNA. We use this sequence of nucleotides in the RNA as a code. And we're going to read these guys, and each of these sets are going to tell us one amino acid, the one that goes precisely in that spot. The central dogma of biology, they're just saying DNA is transcribed into RNA and it is translated into protein. That's normally true, but then you get things like HIV, where they use RNA to make DNA, and then they make DNA to make additional RNA, which makes the protein. But for the most part, this is true for most organisms, except for those retroviruses. We kind of talked about genotype and phenotype. Genotype is just what you have in your genes, but not all genes are turned on at the same time. They're not all active. Some of them are suppressed. Some of them, if you have, say, if I have genes for brown eyes and I've got genes for blue eyes are recessive. The only way I get blue eyes is if I have two blue-eyed genes. But if I have anything else, two browns or a brown and a blue, then this is the guy who's going to be expressed and this guy is going to be recessive. So just because you have the information there doesn't mean that it actually is put into effect. That is the phenotype. That's what you actually are, your physical features and traits. So your genes can have a lot to say. And there are a lot of disease processes and developmental disorders that can derail the expression of these genes. And that's a lot of our genetic disorders. Genotype determines phenotype, right? I mean, I'm not going to have orange colored eyes. Why am I not going to have it? Because nobody's got that in their genotype. So it has to be in the genotype to be expressed, but just being there doesn't mean it's going to be used. Okay. So genotype, if circumstances are one way, if we have one condition, then you might express one phenotype. And if you have a different condition, you might have an entirely different phenotype expressed. We saw that when we were talking about Serratia maricescens. about how it appears red at one temperature, but it will be kind of a white colored colonies at a different. It expresses its different genes at different temperatures. And that's just one example of how that works. So here's our genotype, the DNA, 5 to 3, you see. Well, what we're going to do is we're going to open this up. We've got to split the double helix. And then this is going to be the complementary strand to this. This is the one that we're actually going to use. So the RNA is making a copy of this one. Not both of them, but just that one. So then we have an RNA that's complementary. I'm so sorry, guys. It is, I got it exactly backwards. This guy is actually was coded from here. So code this guy. We didn't code from this one. This was our coding strand. Is it always going to be the bottom one? No, there's no such thing as a bottom strand anyway. These could be in two entirely different genes here, one on this strand and one on that strand, coding for entirely different proteins. But in this particular little example, this is the one they're coding from. So you can see the T, the A, C, and then so forth. So this is our messenger RNA, mRNA. It's been transcribed from the DNA. This is the guy that's going to the ribosomes. And in the ribosomes, we're going to read these guys 3 at a time. That is called a codon, C-O-D-O-N. This is, if you think of the DNA or the RNA, if you think of these as an alphabet with 4 letters, right? I'm going to use the RNA, not the DNA. If I have an alphabet of 4 letters, I can make words that are 3 letters long, and that's a codon. So this AUG is always going to code for methionine. That's kind of the way this coding works, guys. DNA replication. This is what we do in the process of getting ready for cell division, either in eukaryotes such as ourselves or even in bacteria. Each new cell has got to have a complete set of the DNA. Now for us, it's going to be 2 sets of every chromosome, because we get one from either parent. For bacteria, they're haploid, they're only going to get 1 set, but they still have to double their DNA in order that each cell starts off with a complete set. Cells have to be able to reproduce, replicate the DNA, and the whole thing about this is, again, the complementary strand. I'm just going to write one up here, guys. Now, we know I do this to transcribe DNA, right? But I can also do this if I want to make another copy. Instead of having RNA making an RNA copy of this, I can actually make a DNA copy. And of course the opposite on this guy. Notice that if I copy this guy, if I get the complement, I actually come up with this. And this guy is going to be the exact same as that. That's all replication is. We're going to have these complementary nucleotides pair up. Now, I'm going to write this up here. There's our nucleotides. You know what? These guys are naturally attracted to each other, but they need to be bonded together this way so you can have the 2 strands. This is where most of the work is going to happen, is in making these bonds right here. And we'll talk about that. Semi-conservative, what does that mean? It means this is an original strand, this is an original strand, this is a copy, and this is a copy. So when you split your DNA like that, you get 1 original, 1 copy in one cell, 1 original, and 1 copy in the other cell. Anabolic polymerization process requires monomers and energy. The monomers they're talking about are the nucleotides, adenine, thymine, cytosine, and guanine, A, T, C, and G. The polymerization is the process of hooking these guys together. That's what polymerization. Okay, let's go down here. So there's the original. They've got these color coded so that the dark blue is the original. The new strands are this green. And as you can see, you do get a semi-conservative model. You've got one original and one copy. And then, of course, if they go and divide again on a second generation, then you have the semi-conservative. Okay, guys, DNA replication. In bacteria, we know we've got a circular chromosome. There's a place called the origin. This is where we start. So we've got to remember the DNA looks like this. Boy, I'm terrible at art. You can see why I kept my day job. But basically what we've got to do is we have to uncoil it, and then we have to open it up just like a zipper. We have to unzip this guy so that we can get new nucleotides in here. That's the whole thing. Now here at the origin, we unzip it and we go in this direction and we go in this direction until these guys meet over here. When they meet here, then they're finished. We've got the entire thing copied, and voila, you have two chromosomes now. So that's what they mean by the origin. Now on eukaryotic DNA, there are multiple origin points because our DNA is linear, not circular. DNA polymerase, if I'm going to make a polymer, I probably need something to make it with, and this is the polymerase. This is the enzyme that replicates. And remember, all I'm really talking about is to bond these two adjacent together. That's all. Because the A is always going to have those hydrogen bonds with the T over there and with the A. And these are already bound together because this is the original strand. Strands are anti-parallel. So if I've got a strand of DNA and it's 5' on this end, then it'll be 3' here, but the other strand will be just the opposite. Be 5 there and 3 here. That's what anti-parallel means. These guys are also going to have to be synthesized differently because these guys can only lay down. The DNA polymerase can only work from 5 to 3. Now, this is kind of confusing. There's the 5 and there's the 3. But what you have to understand is this is the 5 on the new strand that's being synthesized. So the DNA polymerase is going to work in this direction. But the problem is these guys are anti-parallel, so I can't work in the same direction. So I'm going to have a leading strand, which is going to be really simple because I just start and keep going until I get to the end. The lagging strand, I have to go and make a copy or join together the nucleotides, and I got to jump here, and then I've got to jump here. So what we're doing is we're making one here, and then we have to jump all the way back here and do this instead of just going straight forward like that. Maybe it'll look a little bit easier on here. So here's our replication fork. We've got the DNA unwound. We've got it straightened out. It's no longer coiled, and we've got them separated. And as you can see, these new nucleotides, what they're calling a triphosphate nucleotide, are coming on here, and they're just being attacked. These guys are naturally going to attract to their opposite. What this is the DNA polymerase. What he is doing is he's binding together all the A's and the T's and the C's and the G's on this one strand. Now notice he started in a 5' end and he's going this way. He's going to keep going until he comes to the end. It's really easy for him. This one is not easy. This is the 5, and so this would be the 3' end of this guy. So what he had to do is he had to come here, make this piece. He had to jump back here, make this piece, jump back here. That's what's going on, guys. This is the lagging strand. And by doing that, he's going 5 to 3. He can't go the same direction because that's 3 to 5, and that don't work. He can't go this way because we're unwinding the DNA this way. So the only way he can do it is by doing these jumps. And those fragments are called Okazaki fragments. Okay. Now, there is something else that I haven't told you about yet because I want to make sure we don't load it in too much. Turns out that DNA polymerase has to work 5 to 3, but it also cannot start with nothing there. In other words, it can't initiate. It can only attach to something that's already there. Wow, that's kind of doofus, right? This is the guy that makes these polymers, but he can't start. He can only attach to something that's already made. Well, yep. So what we do is we have something called primase. This guy comes and actually puts an RNA section down, and then the DNA polymerase attaches to that and goes on its merry way. Later, we're going to remove that RNA primase or primer, and we're going to replace it with DNA. There's an enzyme that does that. So every time. Now the Okazaki fragment, you can see that's another DNA polymerase. It's actually number 1 that removes the RNA primer and replaces it with DNA. But every time this guy starts, he has to have an RNA primer. So he's got one there. He had one right there. He's got one right there. When he gets down here, he's going to need an RNA primer as well. So DNA polymerase 3 is going to join the DNA segments. DNA polymerase 1 is going to replace the RNA primer with DNA. DNA ligase only works on this lagging strand. So on the lagging strand, I'm going to have all of this. Okay, let me connect these guys together, show you what I'm talking about. So we've got all of these guys. And what DNA ligase does is he comes in and he joins these guys together. He joins all of these separate Okazaki fragments. If this sounds like it's a really complicated way to do it, well, yeah, but it's the way it is. So there's not anything we can do about it except okay. Now, that's what you need to know. Now, as far as triphosphate nucleotides, I think I may have already told you. So adenine is, we've got a nitrogenous base, we've got a sugar, which in this case is deoxyribose, and then we have a phosphate group. So if this were adenine, this would be AMP, adenine monophosphate. But what actually comes to be bonded there is ATP. We actually we break off those 2 phosphates and we use the energy from that to run that chemical reaction that DNA polymerase does to join these guys together. How does he join them together? Dehydration synthesis. That's the way the entire biochemistry runs. I mean, there may be exceptions, but I haven't run into them. Okay, comparing eukaryotes with prokaryotes. Genome structure, chromosome 1 multiple linear, 1 chromosome origin. We talked about that, multiple in the eukaryotes. These good guys can do 1,000 nucleotides a second. Ours can only do 100 nucleotides per second. Ours is more complicated. No telomerase present on the ends. We've got it in the eukaryotes. RNA polymerase removal, DNA polymerase 1. In eukaryotes, we use RNA ACE H, just another enzyme. We use DNA polymerase 3 in bacteria, and we use pol sigma and pol delta in eukaryotes, but they pretty much will do the same function, guys. Very similar. We use 4 polymerases, have lots of origins. We use shorter Okazaki fragments. And in animals and plants, we put a methyl group, CH3, on cytosine only. We do not put them on the rest of the nucleotides. Okay, let's go into a little bit more detail on transcription. The types of RNA that we can get from DNA is we get the RNA primers, which we talked about. That's used for replication. We get messenger RNA, which is to go out and to the ribosomes and give them the recipe to make that protein. We have the ribosomal RNA, which is, along with the protein part, is actually what the ribosome is made of. We have the tRNA, which, remember, are these guys shaped like this. And these guys have the anticodon on here, and they carry a single amino acid of whatever type. I'll show you some better pictures coming on down here. Okay, all of this is going to happen in the nucleoid region in our prokaryotes. We're going to have 3 steps: initiation, elongation, and termination. You start, you stop, and you keep going until you get to the stop. It's really that simple. Initiation, elongation, and termination. In eukaryotes, we do the RNA transcription in the nucleus because that's where the DNA is. You get some transcription in the mitochondria because, remember, our mitochondria have a separate DNA. Chloroplast in the plants. We have 3 types of nuclear RNA polymerases, not something we're going to be into. We got transcription factors, and the mRNA is not ready to go after it's transcribed. After we make the copy, it still has to be edited. We're going to have to put caps on it. We're going to have to put poly-A tails, which is a bunch of adenines. And we're going to have to do some splicing. We're going to have to cut into it. Here is our eukaryotic. These are the exons, and these guys are the introns. The introns are nothing but spacers. They don't code for anything. Those green ones are the only ones that actually have information we need. So we make an mRNA. There's a 5' cap. We put something on there. We've got an exon there and an exon. Exons expressed. These are the guy. And there's that poly-A tail. So we're going to have to process. We get a spliceosome. And what he's going to do is he's going to actually cut all of this out. and then he's going to take all of these exons and connect them. And now this guy is ready to go. This is a eukaryotic thing only, guys. Translation, where we take those ribosomes, those three-letter words, and change them into a sequence of amino acids. Does it really matter which amino acid goes where? Yes. If you even leave out one amino acid, 1 out of thousands, it can be catastrophic. It only takes the replacement of one amino acid to cause sickle cell anemia. That's all it is, a single amino acid. So in translation, we're going to use those three-letter codons, and those codons are going to tell us which amino acid goes. Now we're always going to, now this is for eukaryotes and prokaryotes. The first codon is always going to be AUG. AUG, almost like in the abbreviation for August. And that is always going to code for methionine. Now in prokaryotes, it's got something called F-methionine, but it's still methionine, just slightly different. In eukaryotes, it's methionine. That is the start codon. So whenever the ribosome sees that, it knows that I start here. We also have stop codons. These guys, whenever the ribosome hits these guys, it is going to stop. UAA, UAG, and UGA. Those are the three stop codons. Do you need to know this? Do you think I would go through it if you did not? Now, you don't have, these are the four I want you to know, guys. These you're liable to see on the test, as well as that codes for this particular amino acid. These don't code for amino acids because as soon as the ribosome hits this, it stops. But if you look, you can see here, and actually we've got all four of these code for leucine. Sometimes they code for 2, sometimes 4, 2, 4, 3. I mean, it's just, it varies. But there's only 20 amino acids, and if you do the math on here, you'll find out there's 64 possible combinations of four letters in three-letter groups. Now, of those 64, 4 of them are taken up as special, the stop codons and the start codons, and the rest of them have to code for the amino acids. Okay, guys. Again, you don't need to memorize all of this, but these 4, the special codons, yeah, you do need to know that. Okay, let's move on. Guess what? The 3 stages of translation are exactly the same as the 3 stages of transcription. You start, you end, and you keep going in between. It's almost trite. It's so, this is a tRNA. Now, this is the end that attaches an amino acid, one of the 20. That's what it looks like in kind of reality. But we're not concerned about its shape right now. This is the anticodon. So I said the first codon is AUG. So the anticodon for that will be UAC. And the tRNA that has the UAC anticodon will be bringing methionine. And then depending on whatever is next, AAA, whatever, we're going to get the next amino acid that comes in there. Yeah, I like the simplified. But all we really need to know is that we've got an amino acid on this end and an anticodon on this end that is going to base pair with one of the codons here. So here comes the F-methionine. The UAC is going to line up with here. Now, we've got, here's the small ribosomal subunit. You've got an A site and a P site. When the large comes on, you're going to have an E site as well. So AUG is the only one that's going to start in the P site. Everybody else starts in the A site. But since he is the very first one, he's going to start right there in the P site. So here comes the tRNA. He's got methionine. He base pairs up with it. He's got the methionine. Now, what's going to happen next? Look at this one. UUU. We're going to have another one that comes into the A site called, he's going to be a tRNA with a AAA. I don't know what, I'm just going to make up one. Let's just say it's lysine. Okay. Now, what's going to happen is we're going to bond these two guys together. We're going to make a bond there, dehydration synthesis again. There's enzymes in the ribosome that do it. And when we do that, the mRNA stands still and this whole ribosome moves down. And it's going to move down 3 at a time. So as soon as this gets bonded, excuse me, as soon as this gets bonded, this AUG is actually going to be moved to the E site, and the E stands for exit. As soon as it hits there, that tRNA is going to let go, and then he's going to go off and find another RNA or find another amino acid. And so then the new one is going to come into the A site, which would this be the ACG. I'm going to be honest with you guys, this is really one that you should watch a video on YouTube. We've got some on the, we have some of these attached to, excuse me, on Blackboard, but you can look for yourself. Translation. If you could see this in motion, it really helps, I think. Okay, so here's our elongation. There we got the AUG. We see these guys move. AUG is now in the E site. P, we've got the new one. So this guy floats off because he's in the E site. The new one comes in. These guys bond, and then the ribosome moves down 3 more, and this floats off, and so on, until it comes to a stop codon. Okay, termination. When we run across a stop codon, the release factors are going to recognize it, and we're going to have the ribosome break apart into the small and the large subunits again. The mRNA is going to float away. It eventually, well, actually not eventually, it's going to be fairly quickly cut up and recycled. We're going to reuse those nucleotides because we don't want it floating around there making more of this than we actually want, whether we're bacteria or whatever. Now we'll say this, I'm going to draw the ribosomes as circles. We do this, we actually you can actually get that same piece of RNA inside of more than one ribosome at a time. There's R1, R2, and R3, maybe even further. And if you notice, we're making 3 of those. This one is of course further along because the mRNA is coming through it. Or we could do it the other way, it doesn't matter. Oh, by the way, why do you translate it? It's translated 5 to 3. Please remember that. Everything is 5 to 3. Okay, and then the once they float off, the polypeptide is ready, and it may be folded, and there may be molecular chaperones that are involved with it to help fold it. So the initiation starts with the ribosome binds to the 5' end guanine cap. First amino acid is methionine instead of F-methionine. And remember that our ribosomes, we have some of them on the endoplasmic reticulum, and they can actually feed the protein into the inside of the rough ER. Okay, mutation. Mutations, when something goes wrong with the DNA. We change the sequence of nucleotides. It doesn't happen all the time, but it's almost always does not turn you into the Hulk. It basically kills you. Mutations are like 99.99% lethal. They do not give you super strength, they just kill you because they usually interfere with something that's very vital in your life function. Rarely leads to a protein that improves things. But over time, you can actually get this kind of mutation. Probably not on the scale that most of the biologists think it is, but whatever. Okay, how can we get mutation? Well, let me draw my first. I'm going to draw 6 of these. So there's my start codon. That's got to stay the same. And here's UGG. And the next one is AUU. Now, a point mutation is where we just miss one particular base pair. What happened if this guy didn't get put in for whatever reason? Now I've got AUG, and now I've got UG. The next one is going to be AUU, but guess what? These guys are read 3 at a time. If I do that, I'm going to start codon, and I'm right there immediately after it, I now have a stop codon. Nothing's going to happen. That's what we call a frame shift mutation. What happens is there's a letter missing here, so this guy, we're going to actually take the next one and consider that to be part of the same word. Substitution, or instead of what if instead of UGG I had UGA there, I would still get the same thing. So either whether I'm actually missing it or it gets changed to a different letter, I'm not going to get the same amino acid. And in some cases like this is called a nonsense mutation. I'm going to have it shortened. I mean, if this stop codon comes too soon, then I'm going to have a protein that's not going to work. Your car keys wouldn't work if somebody took a metal file and filed off the first half of all the little teeth on them. Yeah, the back half is still right, but the front half, it still doesn't matter. It's not going to open your car and it's not going to crank it. Okay. Normal silent mutation. What does that mean? Do you guys remember where we saw that there were up to four different codons for the same amino acid? This is where I change the actual nucleotide, but the change doesn't make any difference. It still gives me the same amino acid. That's called a silence mutation because it doesn't do anything. Yeah, it's different, it's changed, but it makes no practical difference. In this sense, we have a different sequence, mutated template. We get this different. So instead of having alanine as the third, we've got glycine now because of that. We already talked about the nonsense. We get a stop codon way too soon, and so it ends and nothing. These two get totally left off. We talked about the frame shift. If you get an extra one, we talked about if I had one that was removed. What if I get one put in there that's extra? So what if instead of having this, I had this? I would still get a different amino acid because instead of the UGG, now I've got GUG. So this is the frame shift, either a deletion or an insertion, does not matter. It's going to cause a problem. Excuse me. Mutations, what causes them? Well, everybody knows radiation, ionizing radiation, X-rays or the gamma rays. Again, it's not going to turn you into the Hulk. It's going to actually can cause breaks in the DNA and can cause these guys to be changed. And it could actually, the reason it can cause cancer is you've got genes that keep you from going ahead and dividing if the DNA is damaged. Those are called suppressor genes. And then you've got other genes which are kind of like go, go, go to divide. And so if you get these damaged suppressor, you're going to replicate damaged DNA, and that's what a cancer is, guys, simply. Non-ionizing radiation would be like ultraviolet. It's going to cause these dimers. There it is. Thymine will cause thymine guys to bond to each other instead of the A's that they should be hydrogen bonding to. We know that chemicals can cause mutations, can disrupt the sequence in the DNA and RNA, alter the structure of nucleotides, result in base pair substitutions and those missense mutations. And then finally, the frame shift mutations or mutagens that cause these nonsense. There are some things that intercalating agents, ethidium bromide, there are chemicals that we know. Those are the carcinogens you hear about. The chemical mutagens or the radiation, especially these. We all know that radiation is dangerous, and even X-rays have to be done in moderation, but we have more chance, really, I think, of getting involved with chemical depending on where you're at, what you're involved with. DNA, oops, should have jumped that one. DNA repair. Now, there is, this is funny, they misspelled proofreading. Whoever did this didn't proofread their PowerPoint. And apparently I didn't either, huh? Okay, so we can do some proofreading, and we have a DNA polymerase that helps with that. We have a DNA enzyme that's going to cut out the mismatch, and also one to repair those thymine dimers. So there's the ultraviolet, there's the thymine dimer. These guys they're gonna cut them right here, just this strand, and take them off, and these guys will actually, let's go down here. Have to go down this way. Go down here, and all that's going to happen is that these nucleotides are going to bond, not these, but the identical nucleotides are going to base pair up with these guys, and then they're going to get attached to each other in the polymerization process. DNA pol 1 and DNA ligase is going to make those repairs. You can also get a photoreactivation on this. You can actually have light that actually repairs that thymine dimer as well. But that's one of the reasons we have those methylene groups, because it can tell us which one of the, so what if I got a mismatched here? Got an A here and a G. Well, you know what? I got a 50-50 chance of replacing the wrong one. Is the A the right one or is the G the right one? But if this one has the methylene group, and I know it's the right one, so I need to replace this with a T, not replace that one with a C. Whoops, did it again. Sorry about that, guys. I know this is a long one, guys, but can be helped. It is what it is. It's also kind of a tough one, so please study this. Mutants. So any cell that does not repair its mutation and it doesn't actually find itself destroyed by it, or because of its lethality, is going to be a mutant. Wild types are the ones that you normally find. That's the normal cell type. How do you recognize these mutants? Positive selection, negative selection, and then the Ames test. The Ames test, we use bacteria to see if something might be carcinogenic. We measure how fast that this substance causes a mutation in a bacteria when it's exposed to that. And if it's elevated, we may indicate that it's a greater cancer risk, say, in us. Genetic recombination and transfer, horizontal gene transfer among prokaryotes. Vertical gene transfer is just this one cell divides into two and each one has the same DNA as the original. Horizontal gene transfer, this is where a single bacterial cell, I don't know why I'm drawing nuclei in there, guys, I'm sorry. There you go, there's just nucleoid. There's three ways that bacteria can get additional DNA. We've got something called transformation, we have something called transduction, and we have something called bacterial conjugation. These two are oftentimes mixed up on the exam, so please pay attention. Transformation, this is what happens. What happens is we have a bacteria. Remember, let me refresh your memory. We talked about how these bacteria without a capsules could not kill the mice. But if you killed the bacteria that did have the capsule and they're dead and you put them in with the ones who don't have the capsule, these guys get the ability to make capsules. How do they do that? The bacteria has an ability to take up DNA in its environment and make it part of its own chromosome. So if that was the plasmid from this guy that made the capsule, even though he's dead and disintegrated, this bacteria that can't make it is going to take up his DNA, is going to splice it into its own chromosome, and all of a sudden it now has the ability to make a capsule. That's the best example I can think of transformation. I'm taking up DNA from the environment. Now, obviously not all DNA can be done this way, but especially if it's the same species. Transduction, this bacteria gets a virus. The virus will cut the DNA and insert its DNA right there. Now, when the virus gets ready to leave, what happens is it cuts itself out, but when it cuts, it doesn't cut on the same place. So what happens is this virus leaves part of its own DNA and takes part of the bacteria's DNA, and when he goes and infects another bacteria, he actually puts that DNA into that new bacteria. So that's transduction. Conjugation is where these guys make a little tube. Now that's the main chromosome, and this is the plasmid, which, remember what I told you, where they have a lot of virulence factors and a lot of antibiotic resistance, these guys will make a copy of it and pass that copy on to this guy. And these guys may not even be the same species. That's how antibiotic resistance spreads among different bacteria. Okay. Transformation, we get it from the environment. Cells take up the DNA, are confident they have the ability to do that. They have to alter their cell wall and membrane to allow it in. Transduction comes in two forms, specialized and generalized. In the generalized, this bacteriophage, this virus, just carries a random piece of the host DNA to its next victim. Specialized transduction, only certain parts of that DNA can be transferred. Sorry, guys. Okay, conjugation. Physical contact between the two cells. This guy's got a conjugation called the sex pili. I'm not going to read this to you because I've already explained it to you, but you can see the step-by-step. Now, in order for this thing to do this, it has to have the F plasmid from an F plus to an F minus cell. What's happening, we're only getting part, right? Instead of getting both strands, we're only getting one strand, but we know this guy can put the other strand together, right? If once you've got one strand, you can synthesize the other strand easily. Now the F positive, this has the ability to form the sex pilus. Why don't they do their entire chromosome? Well, because these guys will not hold together very long. They're very fragile. Remember, these bacteria are in an environment that's moving around. So generally, this pilus will not hold together long enough to get a lot of DNA. It can only do a very short amount. But fortunately for them, and unfortunately for us, the plasmid is where they carry most of that nasty surprises for us. Transposition, another one. Transduction, transformation, transposition. Transposition, you have certain genes that are called transposons. These genes can actually, let me draw it, can actually cut themselves out of one part, travel around and cut this open and insert in a different part. So that's why they call them the jumping genes. This can cause a frame shift because obviously if we insert new DNA there, we're going to interrupt the sequence of nucleotides and can cause all kinds of unexpected and potentially lethal for us mutations in bacteria. Okay, how do we control all of this genetics? Some genes, I wouldn't say most, I would say some genes are always on because it's just something that just has to be there, right? These are like housekeeping. These things have to be on constantly because life depends on them. Other things, you only need them when you need them, right? Like I don't have a lot of use for my lawnmower in January. Not a whole lot going on there. On the other hand, I don't have a lot of need for my big heavy coat in July. So just like that, these guys have genes that are only used when they are needed. And this allows the cell to keep from wasting energy. Quorum sensing, this is what happens in the biofilms. I think we've discussed this one already, but these guys make these, and if there's enough concentration, then that's going to turn the genome that produces the protein that helps them to form that biofilm and that polysaccharide coat around it. So we can regulate this so that we can stop transcription or we can stop translation, either one. Okay, in prokaryotes, we've got these things called operons. It's a promoter. A promoter is the place where this is the guy that's going to be start here and for the RNA. Here's the genes. The operon is the entire thing. We got a promoter and an operator and then the genes that we need. Now, sorry. The operator is going to be the controller, and these guys are typically, here's a terminology for you, polycistronic. In other words, it codes for several different proteins at the same time. But usually, if it's one that I want to stay off until I need it, I have a blocking molecule here so that RNA or DNA polymerase cannot go by. That's what it is. If it's one that I use all the time, but I want to turn off on special occasions, then it's normally open, and then only when I want it off will I put the blocking molecule there. The inducible operons must be activated, and that's what they call this lactose operon. So normally bacteria don't make lactase, the enzyme to break down milk sugar. Only if there happens to be milk sugar around will they use that. So what they will do is if milk sugar, lactose is available, then that will cause this blocker to remove, and then that will cause this guy to make all these proteins, including the one for lactase. Repressible operons is the one I was telling you about. These guys are always on, and we actually deactivate them by putting that repressor right there on the operator to keep the DNA. The tryptophan operon is an example of that. Well, guys, I think my voice held out. I hope your patience did. Definitely you don't actually have to sit through this entire thing in one sitting. Just do as much as you can and then come back. All right, guys, I'll see you in the next chapter.